sources. I retired a couple of years ago from teaching 32 years at the College of St. Benedict, St. John's University in Minnesota. They're two liberal arts institutions coordinated in central Minnesota. I'm now living in New York City, and I'm a grand nanny to 2 grandkids, and that takes up a lot of my time, but I'm doing a lot of other things as well. I'd like to talk a little bit about my background in modeling. When I was in graduate school a long time ago, I studied Thramming, and the first time I ever used modeling was basically using Frodo on an Evans and Sutherland. I think we just heard about that. It's a $250,000 sort of computer to visualize Thramming. And it was like a religious moment to see that protein that I've been working on for 6 years just stand out. Now, eventually I moved on to, after my postdoc, to undergraduate institution where I did mostly teaching and research as well. So I wanted to incorporate molecular modeling from the get-go. So I used KinImages. I eventually conned the institution into buying me Insight2, which was software that cost about $3,000 a year. And I don't believe they did it, but they did it. They paid that money. And then we used to get magnetic tapes, PDB files from the PDB. Eventually, I migrated to Chime from MDL on the web, and I learned how to do the coding for that. I have very little experience in computer programming, so I'm just a cut and paster with respect to coding. But then, of course, that went out of favor. It wasn't supported anymore. Then, I picked up Pymo and Chimera and just about every other program on the side and found them really stunning. But I wanted something, because at the time I had an online textbook, I wanted something I could plug into there easily and have the students also do. So then I converted from Chime to JMO, which is a Java app. And then, of course, they changed that, when browsers didn't support Java anymore, to JSMO, and I had to redo everything, and I couldn't stand it anymore. And then one day, I was at a meeting and learned about iCn3D. I was at an EB meeting, I think maybe 2019, and just happened to stop by the NIH NCBI booth and met Ronna Morris and somebody else, I can't remember who. And I learned about it and said, wow, this would be perfect for my next project, because not only for the teaching, but I was writing a new biochem book, which I'll talk about, online biochem book for the LibraText project. We'll talk about that later. And I desperately needed something that would allow me to plug these models into the book and at the same time plug rendered models in. So they just weren't floating models in which there was no interactivity associated with them. So I first contacted Yao. It was back in July 23rd, 2019. He's been so helpful in getting me incorporated in the book. I work with Henry Agnew also at UC Davis, who was part of the LibraText projects. He's now gone to UCSD, I think. But they were instrumental in helping me get this incorporated. So first of all, I'd like to talk a little bit about how I've incorporated iCn3D in teaching. Because I've always been a modeler at heart, I wanted to introduce it right away. And we had a very unique chemistry curriculum at our institutions in which we got rid of the standard courses completely. There was no Gen Chem, organic, physical, and analytical. Rather, we blended basically inorganic, organic, and biochem into all of our first 3 courses and labs. So the very first course, 4 courses actually. So the very first course was introductory chemistry. And I was really the only biochemist in the department, but it was critical to introduce proteins and nucleic acids, in part because they're great illustrative examples for non-covalent interactions. So we have a small molecule, a ligand binding to a macromolecule like a protein or DNA. I mean, we used to call them intermolecular forces, but non-covalent is attractive forces that will allow these interactions to occur. So I had the students do this. And one of the things I had them model, I just gave them simple instructions to how to use iCn3D. And then the first thing we modeled was hemoglobin, sickle cell hemoglobin, to show the dimer in which, the mutation from a glue to a VAO at position 6 in the beta chain led to basically dimer formation and ultimately fiber formation. It was so easy to use, just so easy. And then, of course, I would give them basically PowerPoints in which they'd cut and paste images. So they'd have to put various renderings into this as part of their assignment. And then they would look for specific interactions while using some of the techniques that we just heard about, hydrogen bonds, halogen bonds, etc., which we studied now in intro chemistry, but we use biochemical examples in context. And they would literally paste those in here and allow me to sort of assess them. And so that's what we did. I then got associated with the group led by Shushi Dutta out of Rutgers in protein data bank. I worked on a grant with her, which she submitted, and which has been expanded since then, to develop molecular case stories, in which, teaching 32 years, it is clear no matter what software program I use, students struggle with non-covalent interactions and what they really mean and the significance, and presenting them in the context of something that might grip them. The idea of a case study typically having to do with a disease that manifests itself, and it was due to specific perturbation of non-covalent interaction. And so obviously sickle cell hemoglobin is one of the classic ones. So we wrote up, and now there's an actually website that has molecular case net. The idea is to get people to use PDB structures to interrogate both biology and chemistry, bring them together to explain structure, function, and the manifestation of that in human disease. So there's all kinds of stories other than Nicholas's story. And frankly, after 32 years of teaching hemoglobin, I was a little tired of it. But there's all other kinds of interactions for which case studies have been developed. The idea was, being at a liberal arts college when teaching was so important, it was really interesting to see how biologists would teach structure versus chemists. Chemists were afraid of the biology. The biologists were afraid of chemistry. So the importance of actually bringing them together and working at the interface between the two to help them look at the molecular structure, molecular sequences, how that leads to function, and looking at the various forces within a molecule, in between molecules, and the environment becomes really important. And that's where the molecular case net, I think, came in really handy. So in that first class too, I started having the students do 3D printing. And at the time I don't know if iCn3D could do that at the time or prepare the necessary files. I had to use Ligand Explorer from the PDB and Chimera to create the necessary files. But the idea that not only could they visualize this on the computer, but they could actually hold it in their hand. So I would divide them into groups. They'd be assigned a specific molecule, protein typically, or DNA that bound to specific ligand, with the hopes that they would really get a better understanding of the non-covalent interactions between molecules. And this worked in some cases phenomenally well. I had a student, for instance, that decided to print. I think he was working on dihydrofolate reductase and methotrexate, and he gave it to his mom for her birthday, because she was being treated for cancer, and that's what they were using. I had other students that told me it didn't help them at all in understanding non-covalent interactions. But that's the complexities of dealing with undergraduate students. I then introduced iCn3D into my upper division classes, including a specific class in signal transduction. I was using a book by Lim and Meyer called Cell Signaling, created a workbook, and I was going to work with them on getting this published, but other things came in to prevent that at the time. But there was, like, obviously in signal transductions, there's millions and millions of interactions, and being able to show those, not just read about them, but show those and develop these iCn3D links, having the students do it, and essentially using their links as a sort of assessment tool was really important. We then actually did some workshops in iCn3D, and Jiao knows about all this stuff because I was telling him all about it. But through Molecular CaseNet, we held a one-week workshop in July 2020, in which I had one afternoon, I think, to do a workshop on iCn3D. And this is what I would say to the participants of the workshop: that why did I switch from all those programs to iCn3D? One is, my mind can't keep hold of all the commands. If you don't use a program and you come back to it for 5 months, it's like, what were the PyMol commands? Even if I had my cheat sheet, it was, I still struggled sometimes with it. But with iCn3D, it's all graphical user interface. You didn't have to script it. You could write scripts, but I found with time, I'm not even going to do that because I could do everything through the commands that were available through the GUI. You could save the renderings, and this was alluded to before, as a shareable link. I don't think the other programs like Chimera, for instance, or PyMol, you can save session files, and even with JSMO, I think. But to be able to save it as a link which essentially you could email to, like, say, the instructor or to a colleague, and then that link becomes basically usable by those people, one, for assessment to see if they created a rendering that was required of them, or whether you could further change the rendering for this, the student could, and submit it as another assignment. So this was hugely important, the fact that we could save rendering states and shareable links. I was told at that meeting that iCn3D is written into law and it's going to exist forever. You know? And that was, now, probably not true, because who knows what lasts forever. But that was so important to me after the idea of having to switch software so many times. And then, as was talked about before, it's connected to so many databases of information. So it's not just the XYZ coordinates, but all the, like, the active site information, the domain structure. It is phenomenally useful. So if you think about the learning goals that you want for students, or even in a workshop, for the students, you want to be able to find and use relevant PDB files. You want to visualize structures with ready-made scenes or make your own, make measurements, find and render things like domains, annotate them, label them, and also address whether the student can understand what they're doing. So iCn3D to me is like, I wouldn't go back and use anything else routinely. Now, one of the disadvantages is it's not as pretty as Chimera and PyMol, but prettiness doesn't get you, it might get you some places in life, but not necessarily where you want to go. I think it's so important to be able, the images are great, but not spend all your time trying to create publishable quality maybe, but the images that will inform the viewer or inform you if a student knows something about what they're doing. I'm also, as Sandra talked about, I'm also part of a group called BioMoViz. And what we've done is work, and we got funding through the NSF to develop assessments to see if, because when you present, really, this is just information. It's, well, linear code of amino acids and positions of atoms in XYZ space. When we render them, what do the students really understand when we show them these images? Implicitly, we understand them because we are presumably experts in the field, but we need to make explicit what we implicitly know, and then to the students about what's important, teach them the key learning objectives with respect to visualization, and then assess them on that. So we developed a rubric called the BioMoViz framework, in which we have 13 overarching themes. Each one of these, by the way, this PowerPoint is in the Slack channel, so it's available for everybody. You can click on each one of these and you will see a whole list of learning goals associated with, for instance, molecular interactions, or let's see, or alternative rendering. This is a popular one, and then objectives underneath that. So the idea is you find an objective, you find a model that fits that objective from a teaching point of view, and then you find an assessment or create an assessment that targets whether the student gets that. So anyway, and we've created a portal in which we're vetting and creating such assessment questions for users. So, again, you've seen iCn3D. This is an older slide, but it's so easy. You got the viewing window, you got these drop-down windows. So I won't go into any more detail about it. For our workshops, BioMoViz workshops, and for the one through molecular case nets, we'd have a specific modeling skill. Like, for instance, the user needs to load a structure, render it differently, and save it. So we give a particular file, a PDB file. In this case is a low molecular weight protein tyrosine phosphatase, and we go through the specific steps. I was in charge of doing the iCn3D tutorial, so I created this PowerPoint tutorial, and it just walks them through a whole series of things to expand their understanding of iCn3D. And then we would have them literally put screenshots in or PNGs, save their URL, and give us little tips about maybe ways to improve iCn3D, or more likely improve the tutorial that I wrote. And again, here's another example of a modeling skill: display the molecular surface of a protein. And, you know, you can think there's all kinds of different molecular renderings of a protein. What do they mean? And there's some information embedded directly in iCn3D, but the tutorial talks a little bit more about that and talks about ways to style things like glycans, side chains. So now I want to really switch focus of this talk to my book. I've created, along with Patricia Flatt, a brand new online biochemistry text. I had one for decades. I wrote, and I eventually didn't use a textbook, actual textbook in my class, but this is a much more expanded text that essentially can be used for anybody, free, to teach anything you want, but it covers a whole year of biochemistry at a fairly rigorous level. I just want to briefly, I think the easiest way for me to do this, I just want to briefly go back and look at LibraText. I don't know if you're familiar with LibraText. It's an initiative basically developed by Delmar Larson at UC Davis, in which he was awarded a grant, and I've been fortunate to be on several of his grants. And the last grant from the Department of Education was to create online educational resources, specifically textbooks, for every chemistry course in the chemistry curriculum. And that goal is now met. The biochem book will be ready. When I say ready, it'll be essentially ready in September. So there's so many, these pages have so many hits. Then the phenomenal amount of money for a biochem textbook. Students are saving bucket loads of money when they actually when the instructors adopt a LibraText. So this is what my LibraText looks like. And again, it's myself and Patricia Flatt. It's divided into four, I hate to call them volumes, but I'm going to four parts. And it's a classical book in that it's not a POGO, it's not a process, process-oriented guided inquiry learning or problem-solving format. It's a classic text, so it's hugely expansive, but faculty interested in adopting it can pick and choose whatever chapters they want, make their own subtext out of it, and use it any way they want. It's divided into four sections: fundamentals, structure, and catalysis, just like almost every standard biochem textbook. And then bioenergetics and metabolism, again. And then part 3, information pathways, DNA, RNA, proteins. And because I felt like I'm writing this book, I wanted to basically talk about really anything I wanted to talk about, and I didn't have the constraints of a publisher. So for instance, this is this main page for the book. For instance, fundamental special topics in biochemistry. You might look at Schrodinger's cat and wonder. I have a chapter on abiotic origins of life. I've got a chapter coming out soon on climate change and biochemistry. And I'm going to write one and actually seeking help on writing a chapter on quantum biochemistry, not at a super intense level, because students wouldn't be able to, I don't know, the background of most students wouldn't allow that. But again, part 1, here's part 1. It has the traditional sections that you would expect of any classical biochem book. And it has a capstone at the end of biosignaling, signal transduction. So it's super typical of most books. And part 2, again, it's basically everything you would hope to see in any textbook, biochemistry. Now, what would make people adopt a book like this? Well, for an interactive book, for a textbook online, you want it interactive. So for me, that essentially meant two things, including lots of molecular models in which the students could interrogate them and see rendered structures that have been pre-made, but changeable by the student. So these are the different sections I have that contain, at present, all my iCn3D models. And here's a link that can take you them. And again, this PowerPoint is, and the book is freely available right now, the link, so it's in the PowerPoint. But for instance, I got 94 on protein structure, but only 18 on carbohydrates and glycobiology. Well, in part because there's not as many structures with glycans in them because of their inherent flexibility. And I also think there's an inherent bias toward proteins. So anyway, this is the distribution I have, and I have a huge number of modeling, like iCn3D models on signaling as well. So let's see. So let's see what my next slide takes us. I guess here I am. So this is what I would do is, and I'll show you some examples in a second. I wanted to actually have the molecule appear right in the text, but that would mean at any given chapter, I might have like 15 models in which the students could rotate them. And it soon became apparent that even with iCn3D, I couldn't have them directly incorporated. I had to have links to them. So what I did is I would create the model. And again, you saw how you, and I'll show you how to do this in a little bit. You can simply get the code that, you remember you saw how you could get a short URL and use that in a browser to re-render the molecule. You can also get a list of commands that you can simply paste into a web page. And what I do is I have a separate web page, one page per one molecule. And then in my actual chapter page, I'll have a PNG of the molecule. You can click on it and you will get a pop-up of the molecule which you can interact with. And I also have a second link directly that goes to iCn3D, and I'll show you that in a second. And it's been, again, made so easy by Zhao and Henry Agnew at UC Davis, just creating a little template for me in which there's some Decky script, and all I would have to do is plug in the information directly off of the iCn3D. So here are some examples of what I've done. So and remember, there's almost 400 of them, and I also have to say, okay, when you write a textbook, there's no way, first of all, that I know enough biochemistry to write a textbook on every field. I've come to realize I don't know anyone that would, because it's such a dynamic and huge field. So you do the best you can. And of course, I have my own filters, my own lenses that I use in terms of what I like. So my time, it's taken an enormous amount of time. So all of a sudden, okay, here's a protein model, it's a DNA molecule RNA that I've got to use an iCn3D model in the book. Well, I've got 400 of these to make, so I cannot spend 100 hours creating the most exquisite model to put into the book. I have to work quickly. So the models in the book are, and I learned about I learned how to use iCn3D over time. I started this like 4 years ago. So my models now are probably better than they were back then. But anyway, and also I'm not an artist. So, and I say this because I feel like I can use modeling software to be artistic. At the same time, sometimes my colors combinations might be artistically not pleasant. Let me show you a couple. So I'm going to go to this, the sugar link, the sugar-coated lectin decoding. So this link will take directly to chapter 7.4 in the book. And here's an example of an iCn3D that I've embedded, and it's the simian immunodeficiency virus GP120 core protein. This is the PDB code. Now here's a link that could take me directly to the ICN page. If I click on this, I can pop up this and see an image, and it'll be the same image that you just saw, only this is obviously rotatable and you can interact with it. The surface I just made white because that's not what I was interested in. The carbohydrates are, and you can even use glycan cartoons. You know, there's a standard nomenclature for showing glycans. And even this image here is modifiable. So if you click the menu, you can go down to, let's say, File, Share a Link. Okay, you can share a link, and again, you get the short link and pop it into a new window, or this is the script I just showed you before that I popped into the template I had. So it's really super easy to see this and to use it. So I decided to put two links in. Okay, let's see what's going on. Oh, what do I gotta do here? Let's get rid of this. So I would always put two links in that would allow students to have the full iCn3D, well, again, with the If you go down to analysis, you can see defined sets. My defined set should be shown as well. And again, you can interact with this in any way you want. You could, for instance, let's just turn off. Let's say we don't want to see the surface anymore. We can remove the surface, and the surface is gone. So you can interact with these. I try to render them in ways I thought were most pedagogically useful. That doesn't mean I succeeded, but you get the idea. Let's see, here's another one. I'll just go directly to the external link on here. I think this is just a nascent HDL particle. This is actually a PDB file. You can't really get PDB structures for lipoproteins. But anyway, again, you have this image, and now you see a lot of lipids embedded in here too. And if we go again to, I have to move this. Let's see. I want to go to analysis. I want to move this. Pardon me for a second here. I want to go, let's say, style protein. What if I wanted to change that to sphere? Well, I didn't select anything, so I have to do that. So if I go down to predefined sets, I have presumably here, let's just pick the protein. Now what I can do, for instance, is style. I could change these at will. So how about style protein sphere? Okay, you can do that. You can change coloring. You can make surfaces. So now let's say I want a surface type. I want to make an electrostatic surface, so I can go to analysis. I'm showing you some of the things you can do. Let's go to Delphi potential. And let's say I want a surface with potential, and I want to show molecular surface, and I want to show this. And it'll pop up in a little bit and show that protein color-coded based on molecular electrostatic potential, with red being the most negative potential and the blue being the most positive. So, again, it's like, what do you want the student to get? I mean, target what you want, show what you need to show, but have them be able to interrogate the structure so they get a real sense of what it looks like. There's so many cool things you can do, like membrane proteins. I'm going to pop up this just to show you a couple. Because here, I mean, you can show the lipids too. And, if you're looking at a peripheral membrane protein, I mean, why do you need PyMol? Why do you need Chimera? I mean, this gives you everything that you'd ever want. And it's so easy to use. So I wanted to just show a couple one of these. Here's another one. This is the long load time, but it's kind of fun to see. So is there any questions why this is loading? Is this going to show a dimer of a particular enzyme in glycolysis? This will pop up in a second. And I color coded this two surfaces differently. And you can see, I think, the interaction showing. And again, there's an infinite number of ways to render these things. So it turns out picking the one that's most relevant is just a matter of taste at the moment. I'm going to wait, probably because we're zooming. It takes a while, so we're just going to wait because surface renderings typically take a little longer. And maybe what I could do is let that pop up and see if there's anything else I want to do. Yeah, so I could wax and wane about this, but I just wanted to, before I maybe break for any questions, I have done something else in the book. I have added computational modeling, and I never know what group of people would be interested in this, but I think it's utterly important. I have incorporated VCell. It allows you to model simple, so very complicated signaling pathways. For example, you can model very simple or very complex reactions, such as the reversible conversion of A to P. And you can run the analysis directly in the page. I have both embedded graphs in which you can change sliders. You can actually run the V-cell analysis directly in the page and actually see the output. And to give you a final example, so some of you may be interested in that, but this is the rendering, and I think this is actually, I think this is both artistically and biochemically interesting as well. In terms of rendering, I think some of the key things, you can label things, show the active sites in different, actually different parts of the molecule. I thought the last thing I want to do is I just show you this one thing simply because I just think it's a fun thing to show. I think it's good. So this is my unit on signal transduction, cell signaling. I'm going to go down to, I have animations in this as well as static figures as well as models. You can see here's an iCn3D I have. You can also easily superimpose molecules to show. What I'm going to do is I'm going to run this model. It's going to take, this is the map kinase cascade in which I have a map KKK having to become phosphorylated to phosphorylase the secondary map KK, which ultimately gets doubly phosphorylated to finally phosphorylate map K to get the doubly phosphorylated active molecule. But this thing can come back and phosphorylate this in a different site and inhibit it. So I'm just going to run this and now simply ask if there's any questions. And this will take about a minute to run, where like the software will be changed to make it run very quickly. But essentially, that's what I wanted to say. If I had to summarize, one, iCn3D is just an amazing tool. And I have to give great kudos to the developers of this because it does everything you'd want to do. The fact that it's tied into these databases, for instance, sometimes if I don't know an active site, I can just go functional sites and the active site is already there. I don't have to go to the literature and to figure out which amino acids are actually involved in the active site. But the sequence information, the alignment tools, the mutagenesis tool, I think makes it a modeling program of choice. And actually, every time I give a workshop, I swear I get converts. They say, oh my God, this is so great to use. Anyway, this is the output from the simulation. And you can see that you get oscillatory behavior. It's like a wave of signaling kinase that basically vary over time in the cell. So that's the kind of interactivity I have. So the iCn3D and the modeling allow that to happen. So I'm going to stop sharing my screen and be happy to take in questions.